right, future doctors, welcome. Today, we're not just flipping through a textbook. Nope, we are getting to know our enemy, tuberculosis. Think of this as a strategic briefing. In this first part, we're going to dissect this invader, look at its defenses, and map out its initial attack plan. This is all about getting you ready for those exams, so let's get focused.
So you gotta ask, what's the secret sauce? What makes this ancient bacterium so incredibly successful at what it does? Understanding this is the absolute key to mastering this topic. We're gonna break down its tactics, piece by piece, so that no matter what question comes your way, you will be ready. Okay, let's dive in.
First things first. In any good investigation, you have to know exactly who you're looking for. So let's start by properly identifying the organism, or should I say organisms, responsible for TB.
Okay, listen up, because this is a classic exam pitfall. The bug that causes TB isn't just Mycobacterium tuberculosis. If you want full marks, you have to write M. tuberculosis complex. Why? Well, because it's not a lone wolf. It's a whole family, a whole complex of genetically related bacteria that cause the disease. Writing complex shows the examiner you really know your stuff.
And here's the lineup, the usual suspects. Now, M. tuberculosis is our main player for sure, especially in humans. But knowing a few of the others, like M. bovis, which you can get from cattle, really shows the examiner that you've got that extra depth of knowledge.
Right, if you want to defeat an enemy, you have to understand its defenses. And let me tell you, M. tuberculosis is walking around with some of the best armor in the microbial world. We're talking about its cell wall. This structure is the key to its survival and, you guessed it, a very high-yield topic for your exams.
Okay, take a look at this. This right here is the blueprint to the Bacillus fortress. We're about to break down these layers because once you understand the structure, you'll understand its most famous party trick, being acid-fast.
So how in the world are you going to remember all these layers, especially in the right order? Simple. We've got a mnemonic for that. Just remember PAM. Peptidoglycan, arabinogalactan, mycolic acid, and mycosides. PAM. Now the absolute rock star of this group is mycolic acid. These are long-chain fatty acids that create this waxy, almost waterproof barrier. And that barrier is what gives it its acid-fast property. It's why the bacterium holds onto the stain even when you wash it with acid. It's a key diagnostic feature. So remember, PAM.
Now, hidden inside this armor is a secret weapon. It's called Lipoarabinomannan, but let's just call it LAM. Think of LAM as the bacterium's invisibility cloak. It's a key molecule that allows the bacillus to get eaten by our immune cells, our macrophages, and then survive inside them. It's incredibly sneaky, and we're about to see exactly how it pulls this off.
Okay, so we've analyzed its armor, we know about its secret weapon. Now let's track the invasion itself. How does this thing get from the air all the way down into the deepest part of our lungs?
So it all kicks off when someone inhales these tiny airborne droplets. Most of the bacteria get trapped on the way down, but the really small ones, less than 5 microns, make it all the way to the alveoli. And that's where its secret weapon, LAM, comes into play. It helps the bacillus stick to a macrophage, which, doing its job, gobbles it right up. But, and this is the brilliant part, once it's inside, the bacillus sabotages the macrophage's internal disposal system. It literally blocks the little pouch it's in, the phagosome, from fusing with the lysosome, which is full of digestive enzymes. So it basically prevents its own execution and turns our own immune cell into a cozy little safe house.
So once the invasion is detected, the alarm bells go off and our immune system launches a counterattack. But here's the thing. The outcome of this battle can go one of two very different ways. Understanding this duality is absolutely critical for your pathology exams.
That infected macrophage doesn't just sit there. It starts waving a red flag. It takes little pieces of the mycobacteria, these antigens, and presents them on its surface. It's basically screaming for help. And who answers the call? The special forces of our immune system, the T helper cells, specifically the Th1 cells. They are the commanders of this whole operation.
And right here, this is the crucial fork in the road. The immune response can go one of two ways. On one side, you get a good, strong response that activates macrophages to kill the bugs. Let's call them the containment crew. But if the immune response is weaker, you can get an over-the-top tissue-damaging reaction. This is the demolition crew. The patient's fate really hangs on which one of these responses dominates.
So let's look at the good outcome first, the containment crew. The Th1 cells release a powerful signal called interferon gamma. You can think of this like a shot of adrenaline for the macrophages. It supercharges them, gives them the power they need to finally kill and digest the TB bacilli. Boom, job done. The infection is contained. This is what happens in most healthy people.
But what happens if someone's immunity is weaker? The body tries a different, more destructive tactic, a delayed-type hypersensitivity reaction. This is the demolition crew. Instead of a controlled kill, you get widespread damage to the lung tissue itself. And here's the crazy paradoxical part. This destruction actually helps the bacteria. The damaged tissue liquefies, which frees the trapped bacilli and gives them a perfect environment to multiply and spread. The body's own defense ends up working for the enemy.
All right, let's rewind a bit and go back to that successful containment response. When our immune system manages to corner the invaders, it builds a tiny prison around them to keep them locked up. This little prison is called a granuloma, or a tubercle, and it is the absolute hallmark of tuberculosis.
Now, these granulomas aren't static. They evolve over time. At first, you get what's called a hard tubercle. It's just a solid ball of immune cells, macrophages, lymphocytes, you name it. No dead tissue yet. But later, the center of this ball can die off. This is called necrosis, and that turns it into a soft tubercle, which has this dead, necrotic core. This progression from hard to soft is a key feature you've got to remember.
So what does this dead stuff in the middle look like? Well, pathologists have a very famous and kind of gross description for it: caseous necrosis. The word caseous literally means cheese-like, because the material looks like soft, crumbly cheese. Yeah, it's a vivid image, but trust me, it's so descriptive you will not forget it come exam time.
Now, this cheesy necrotic center is a really nasty place to live. There's very little oxygen, the pH is low, it's a totally hostile environment. And since TB bacteria need oxygen to grow, this basically puts them to sleep. And that's the key. The bacilli aren't dead, they're just dormant, lying in wait. A fragile truce is called. This is the basis of latent TB, and these bacteria can stay dormant for years, even decades. So we've reached a stalemate. The enemy is contained inside the granuloma, but it is not defeated. The bacilli are just sleeping, waiting for a chance to reawaken. Which brings us to the big question. What happens when a person's immunity drops and this truce is broken? Well, that is exactly what we are going to cover in Part 2, where we'll talk all about clinical features, diagnosis, and what happens when this sleeping giant wakes up. I'll see you there.